So I was invited to talk about the Immunoscore and the role of immune cells against cancer. So my lab has been focusing on trying to really analyze deeply the tumor microenvironment and to really understand what are the important cells within the tumors that either promote or control tumor progression. So the reasoning behind it was first, when I started more than a decade ago, the field of tumor immunology was not at all recognized. Most of the studies were focusing on tumor parameters, tumor genotype, and I thought that the immune system was important as well. And so I've tried to analyze the complexity of the tumors in total, so not only the tumor cell complexity, but also the tumor microenvironment complexity, that is the complexity of the immune system infiltrating the tumors. And so that was with the idea that patients having long-term survival versus those having a very short-term survival of cancer, one of the reasons could be because of the immune system of the patient and of the immune response of the patients. And it turns out that indeed subsets of immune cells are extremely important to predict long-term survivors in the patient's survival. These are the cytotoxic T cells, the memory T cells with a memory capacity. And so we have shown that over the last 10 years. No, we looked at every parameters related to the immune response. So not only the cell types, immune cell subpopulations, but also the receptors that they are expressing, the factors that are secreted by those cells or that are present within the microenvironment. So cytokines, chemokines, all sorts of immune-related features. And some of them are very important. So the main cells which really predicts the survival of the patients are the adaptive immune cells, so especially the T cells, the cytotoxic T cells, the memory T cells, and recently we also have shown the importance of the T follicular helper cells and the B cells. So all those adaptive immune cells are very important. So together with other immune related molecules like the Th1 type of orientation, so interferon gamma, TBAT transcription factor, the gamma cytokines, the cytotoxic molecules such as granulizine, perforin, granzymes are very important, and some chemokines are also very important. So what we did was really to have a very deep analysis of the tumor microenvironment, and basically we took the most powerful cells to predict patient survival, and we put them into the Immunoscore assay. So the Immunoscore assay is a very simple test, but still very powerful to predict patient survival and to classify cancer patients. So it's basically the densities of cytotoxic and memory T cells infiltrating tumors, taking also into account not only the density of these cells, but also their location in different tumor regions. And basically with these information of the density of these cytotoxic and memory T cells, we give these Immunoscore results that allows us to classify cancer patients. So it can be done in a routine pathology lab settings with regular paraffin tissue from tissue tumors embedded in paraffin, and then it's two simple stains with the two antibodies specific for the cytotoxic and memory T cells. And then it's a full quantification of those cells. So it's an image-based analysis that is done with digital pathology. So it's not a subjective selection of a particular field, the whole, it's an objective quantification of the whole immune cells infiltrating the tumor. So we have tested it in multiple cohorts of patients with colon cancer, with rectal cancer, with breast cancer, at different cohorts at all cancer stages, so from stage 1, stage 2 to stage 4. So the Immunoscore is powerful at any stage, at all stages, even the earliest tumor or the latest tumors. We have ongoing studies in colon cancer with a consortium that I initiated, an international consortium with 23 centers in the world currently testing this Immunoscore. And also we have a national prospective study in France ongoing involving 6 hospitals in France. So, well, it's going to be tested based on the disease-free survival and overall survival of the patients. And what we have shown in the past and what others have shown also is that the density of these cytotoxic and memory T cells, and especially the Immunoscore, that is the most powerful way to quantify them, is extremely powerful in terms of classifying cancer patients and allowing their prognosis. So the interpatient variability, so yeah, it means that some have a very high Immunoscore, so strong T cell, likely T cell priming, a lot of cytotoxic effector cells, a lot of memory T cells. And those patients will do well compared to the patients not having these T cell infiltration. What we have also shown more recently is that, in fact, initially the tumors, the density of the infiltrating immune cytotoxic and memory T cells is very high, and with immunosuppressing mechanism, these densities are decreasing over time, and when they are decreasing, the tumor is progressing. So we have a relationship between the density of these cells, so between the Immunoscore and the tumor progression and invasion parameters. So hopefully, so today there is not a single test to evaluate the immune system of a cancer patient. Nothing is done in routine, and hopefully the Immunoscore would be the first standardized, reproducible, powerful immune test that could be introduced into the cancer classification. So this would be an immediate benefit. The second is that it may completely change the picture and change the way patients are treated. For example, there are patients at, let's say, late stage, having like stage 3 or very aggressive chemotherapies. And we know that some of these patients with very high Immunoscore will do very well, but probably not because of the treatment they are receiving, probably just because they had this very strong memory T cell to start with. Vice versa, there are patients with Immunoscore 0, so very low T cells, memory T cells in their tumors. And those do very bad, even though they have received aggressive chemotherapy. So chemotherapy is clearly not efficient in those patients. So, and we could also take other examples at early stage tumors. Now we can predict very high risk of tumor recurrence at early stage tumor with a very low Immunoscore. And so it would be wise to start new clinical trials addressing the question of are there any benefit of chemotherapy or other treatment like immunotherapy in patients with these early stage tumors.